Hello, good evening, one and all. Welcome to the webinar, which is on basic immunology. I'm Dr. Sathyabama, Senior Scientist in MTR Labs. I thank the opportunity given by Sri Sai Clinical Research Institute Private Limited. So, to give a small introduction about our company, Microtherapeutics Research Lab, which is established in the year 2005. It's a full service CRO, clinical research organization in Chennai, which specializes in providing bioavailability and bioequivalent services to the pharmaceutical and biotechnology industry worldwide. With a commitment of providing world-class services, MTRL offers customized research solutions tailored to meet the unique requirements of its clients. Apart from BA/BE studies, we also provide services like diagnostics, medical writing, and regulatory affairs. Our achievement include we are well recognized by international regulatory authorities, and we are the first CRO in India to conduct postmenopausal women study for US FDA submission, and also we pioneered in special population studies. We have filed more than 500 BA/BE studies for US FDA, and in which all of them has got approved. So coming to the webinar of this session, it's on basic immunology. We are just trying to cover the introduction part of the immunology, since and everybody would admit that immunology is a vast, extensive subject, and we are giving a basic introduction on the same. So to know the fundamental of the immunology subject, we would like to define three most important things, where the immunology covers the study of all aspects of the host defense. That is, when an infection occurs, what all the aspects of host defense takes place, and also the adverse consequences of the immune responses. When a host is getting infected with the pathogen, the state of the protection which occurs at final stage is called the immunity. So immunity is the state of protection from the infectious diseases. So after this exposure of the pathogens to the host, the immune system, a versatile defense system is developed in the body, which is collectively called as immune system. This includes from invasion of a pathogen till cancer, whatever might be the cases, the immune system or the development of the defense system is the complete immune system we get. So how does the pathogen gets entered into the host or affect the host? Where it should first gain access to the body. Anything to enter, it should have the access to enter into the body. How does this get access is attained by the pathogen is it gets attachment to enter the host cell with the receptors present in the cells. On the pathogen, receptors on the pathogen gets fit into the host cell receptors through a lock and key model. So wherever there is a proper fit, it gets access and it enters the cell. But whenever there is no proper fit or there is no receptor to fit, then it doesn't enter. After getting fit to the receptors, it reproduces by avoiding the host immune system. Host has the largest versatile, as we said, largest versatile immune system developed on its own. But then this pathogen should escape from that immune system to produce harmful changes. So, and there should be such a long time to get escaped from the host immune system. So now you be able to recognize that it's not easy to be a pathogen, right? So there are more steps involved to enter as a pathogen also. So how does this immune system completely works? So this immune system of the host works in two main immune pathways, innate and adaptive. Innate is non-specific and adaptive is specific. This innate immune system, innate immunity is developed again through two line of mechanism. The first line of defense and the second line of defense, wherein the adaptive works in two modes called cell-mediated immunity or humoral, which is antibody-mediated immunity. So the main difference in innate and adaptive is the non-specificity and the specificity respectively. So coming to innate immune system in detail, innate immune system is the immunity which we are born with, that is inborn. So when we are born, there are a specific immunity in the system developed as from the fetus, so which is said to be the innate immunity. But this is not specific to particular antigen, so they are called as non-specific immune which we have developed. And there is no memory. These cells or the immunity which is developed inborn doesn't have any memory, so that when an antigen is encountered on the second time, there is no memory, so there is again no defense for this. Immediate defense will not be available. So this innate immunity works in two again it works in two ways. There is first line of defense which takes place, and there is a second line of defense which it takes place. The first line of defense is inclusion of the physical barriers, chemical barriers, and the biological barrier which is present already in the body. So the first line of defense, as I said, the physical barrier. In the physical barrier, the skin and the mucous membrane plays major role in the physical barriers. Skin, as you know, it is the largest organ of the body, and it is the major physical barrier. The hair follicles present in the skin traps these pathogens, and it inhibits from further colonization of the pathogens. So the pathogen cannot enter since a trap is already available in the skin. And the mucous membrane, which is the outer lining of few organs like nose, mouth, stomach, etc., will have a mucus fluid present in them. These mucous membrane, the fluid will consist of mucin, which is a glycoprotein, and also antimicrobial peptides that also helps in killing of the microorganism. So this to act as the physical barriers. Coming to the chemical barriers, the pH of the system, particularly the skin, gastric acid, the vagina, all have a low acidic pH. You know, the low acidic pH is not an optimal condition of most of the bacteria. So the skin will have pH of about 5.5, gastric acid 1 to 3 pH, vagina will have 4.4 pH. So at this acidic pH, none of the bacteria can live happily, so that there will be no colonization of the bacteria. And next, the enzymes present in the body. Enzymes like lysozyme, peroxidase, pepsin, defensin, etc. are present in our body. For example, lysozyme is present in saliva, tears, mucus, etc. Pepsin is present in gastric mucosal defense, gastric juice. Beta defensin is present in epithelial cells. So all such enzymes will be the chemical barriers of the body to kill the pathogens. And next coming is the biological barrier. Biological barrier is the barrier where the immunes or the host is developed with few of the microorganisms inborn, which is called to be the normal flora. The normal flora, which is now said to be the microbiome, is a biggest or a largest immune providers, wherein they are all beneficial bacteria which secretes more of the antimicrobial compounds, and it also competes with the pathogen and avoids the pathogen to colonize in our body. And recently, interesting research is happening in the microbiome area, which is which gives us more insight or greater insight in the correlation of the immunity developed and the microbes present in them. So these are the first line of defense. And coming to the second line of defense, the second line of defense includes the phagocytosis, complement proteins, inflammations, etc. Phagocytosis. The cells involved in the phagocytosis, and the second line of defense is these type of cells, which includes the WBC mainly, which is the monocytes or macrophages, mast cells, eosinophils, basophils, dendritic cells, NK cells, natural killer T cells, delta gamma T cells, and mate cells, endothelial cells. All these cells in a combination of action, it will play a role in innate immunity. So these mainly relies on pattern recognition receptors. These are the receptors present in the cells which will first detect the antigens, which whatever the antigens that's coming, and they respond through pathogen associated molecular patterns, which is called as PAMs. These are the common structure present in the pathogens. So the key cytokines, where whatever it is released when an infection occurs by these cells, includes TNF, IL-1 and IL-6. Interleukin-1, interleukin-6. This is tumor necrosis factor. So these are the three main cytokines which is released by these cells. So what does it do? It initiates the cell recruitment. These cytokines plays an role in recruitment of other immune cells to come to the area of infection, and then a local inflammation occurs then, so that the pathogens, all the pathogens are cleared from that area. So as I said, the phagocytes are from these cells, the phagocytes are the macrophages and neutrophils. And then dendritic cells, the action will be, they present the antigen to the adaptive immune cells or the other immune cells. So these are called antigen presenting cells. And natural killer cells, they have the enzymes called perforins, granzymes, which will in turn kill the pathogens or the cells which is engulfed. So coming to phagocytosis, the process by which cells engulf the pathogen is called phagocytosis. To simply say, it is eating the pathogens. These cells are capable of eating the pathogens. That's what the process called phagocytosis. So first, the phagocytes has the PRRs, as I said, the pattern recognition receptors, which will recognize the pathogen and bind to the PAMs, like molecular patterns. Then the activated cells expresses whatever the cells has identified the pathogens, it gets activated, or the antigens, it gets activated. So these activated cells will express more glycoprotein receptors. These receptors, what they do, they help them to move to the site of infection, bind strongly with the microbes under the influence of cytokines. So then the cells begin to engulf the foreign particles. To engulf the foreign particles, first this phagocyte, in the sense the cells which is involved, either the macrophages or dendritic cells, it will enlarge its cytoplasm to form a pseudopod. This is called pseudopodium. It has enlarged its cytoplasm to form a pseudopod. This pseudopod, it surrounds the molecule or the pathogen which has entered, or which is present at the site of infection. So this pathogen will be taken into the engulfed, it will cover up this pathogen and take as a phagosome. This will be engulfed inside the cell. The fusion of, with the vesicular structure, the fusion of the pathogen is happening, which is said to be the phagosome. The fused vesicular structure, this phagosome, is transported into the cell, and then there is a lysozyme, an enzyme called lysozyme, which is the digestive enzyme. This gets fused to phagosome and forms the phagolysosome. What this phagolysosome will have this enzyme present in it. So this digestive enzyme, which is the hydrolytic enzyme, digests the phagocytosed particles. Whatever has been phagocytized, it will digest. So these digestion will happen, and the indigested material will be eliminated, or the residual body will be, the digested material will be the eliminated from the cell through exocytosis process. So the phagocytosis has the sequential step: attachment of phagocyte to pathogen, ingestion of pathogen, formation of phagosome, formation of phagolysosome, and destruction of pathogen. Finally, the elimination of debris happens, which the debris is coming outside the cell. Next comes the complement system. This is a more crucial system, a more crucial pathway which provides the innate immunity, wherein this complement system takes place in three major pathways. One is classical, lectin, and alternative pathway. So classical pathway, where the C1q is a protein which will detect this antigen-antibody complex. When an antigen has come, an antibody is also is produced against the antigen, And then when this 2 gets complexed with this binding sites, C1q protein is the protein which detects this antibody, and then this classical pathway is initiated. Wherein lectin pathway, there is a mannose binding lectin receptor which encounters the pathogenic carbohydrates motifs and then activates the serine proteases. Proteases is again the enzyme which helps in cleaving the pathogens or the cleaving the protein structures, the carbohydrate structures present in the pathogen. See, the alternative pathway is activated only when the C3b molecule is C3b molecule undergoes spontaneous hydrolysis. Otherwise, both this pathway, classical and lectin pathway, gets activated and cleaves the C4 and C2. All this three pathway culminates or like forms the C3 convertase enzyme to activate this C3. After this C3 is activated, there might be three types of mechanism to eliminate the pathogen. One is phagocytosis, as we discussed before, or by cell lysis and activation, which takes helps of the membrane attack complex, and then opsonization might also happen, which is through opsonin proteins. Then there is a site inflammation in the site of infection. After this, all this pathway takes place, or all the C3A is getting activated. There is an inflammation in the site of infection. So this is about the complex complement system, in which the phagocytosis and the complement system plays a crucial role in the innate immunity. So coming to the adaptive immunity, whenever the innate immune system is not able to destroy the germs, then comes this adaptive immunity. Adaptive system plays a role in developing the immunity against that pathogen. It will take time to recognize the germs, but than the immune system, but when it does the job, it will be very accurate. It's specific immunity, as we discussed before, and also the main thing is it keeps memory, so that when an antigen is encountered on the second visit, it will act faster and the pathogen is rapidly eliminated from the body. So the adaptive immune system is made up of T lymphocytes, which is present in the tissue between the body cells. B lymphocytes also found in the tissue between the body cells. And then comes the antibodies in the blood and other bodily fluids. So the immunity, if it is developed through T lymphocytes and B lymphocytes, it's called cell-mediated immunity. If the immunity is developed through antibodies produced against the antigen, then it is called humoral immunity. So T lymphocytes, this plays a major role in the cellular immunity. T lymphocytes is named based on its origin from thymus. It's directly involved in destroying infected host cells, producing cytokines and activates other immune cells, and also involves in regulating the immune response. So there are three types of major subset in this population. T lymphocytes is of many subsets, but the major predominant subsets are T helper cells, cytotoxic T cells, T regulatory cells. T helper cells is, as the name suggests, it is going to help in identification of the foreign particles. It will always have CD4 as a receptor or a protein present in T helper cells. And this is going to activate MHC class 1 complex, which is again an immune complex activated to kill or to identify the antigen and destroy them from the host. So class 2 MHC complex is again a vast mechanism, which we shall take it in other webinar. This is just an introductory class, so we are just going through brushing up the immune system. And comes this cytotoxic T cells. As the name suggests, it is going to kill whatever the identified antigen. So it expresses CD8 protein, and this activates class 1 MHC complex, class 1 MHC molecules. So these liberate granules into the cell, which is to be killed. Whatever to be killed, they secrete granules inside them, so that the cells will be killed. Then comes, these T cell CD4 and CD8 T helper or cytotoxic T cells. After this activation, that is after they encounter the antigen, some T helper cells will become memory T cells. So that these T cells will act when the infection is affected on the second time, over the second time. There is a population called T regulatory cells. It is called Tregs. Tregs is a unique population of helper T cells, which is main cells involved in maintaining the equilibrium. Equilibrium in the sense to maintain the homeostasis of the host. This uses various mechanisms, but just to give a overall view of the Tregs, Tregs are unique cells and divided into naive Tregs. Naive Tregs is nothing but whichever the Treg population which doesn't have encountered the antigen. And memory Tregs are already encountered the antigen and have the memory that this antigen has already come into the body. And there are effector Tregs which is going to play, or which is activated Tregs which is going to play the action of whatever the role they are involved in. This Tregs research is intensive recently after this population is identified, and this balances the immune system. So only this has been shown. This tries to balance the immune system by preventing or dampening. If at all needed, it will prevent the immune response not to happen, and if needed, it will give the immune response. So it is specially designed to regulate the immune response, and hence the name T regulatory cells. If there is an imbalance in the T regulatory cells, they might be increased Treg function also will cause problem. Impaired Treg function also will cause problem. So this should be in homeostasis state. Increased Tregs might cause cancer and low response to vaccination or tissue degeneration. And if there is impaired, if the Treg is not functioning properly, also it will lead to autoimmunity or chronic inflammation. So next is the B lymphocytes, which provides the humoral immunity or antibody-based immunity. B cells, as the name has been derived from bursa of Fabricius in birds or bone marrow of human, where the origination was identified. This is antibody mediated immunity, as I already said, and the destination of B cell is plasma cell. These B cell recognizes the antigen by B cell receptor, BCR, which again stimulates the activation of B cell. This receptor, when identifies the antigen, then the B cell will get activated, and it produces high affinity antibodies. As you know, all the adaptive system is going to be very specific. These antibodies developed in the adaptive immunity will also be high affinity with high attachment to the antigen. And it provides long-lasting memory responses for the secondary encounters. These naive B cells, after contacting the antigen, proliferates and differentiates into effector cells. Effector B cells, it's called. Then produces the antibody with antigen binding sites. So they have this high affinity towards the antibodies. So the antibody, this is what I said. The B cell has the receptor. When it encounters the antigen, the resting B cell will become an activated effector B cell, which produces high affinity antibodies in a larger secretion after proliferation and differentiation. So this is the antibody produced, which is Y-shaped molecule with antigen determining sites present in them. It will be 2 antigenic determinant sites present in them. These are called bivalent. These can correlate with other antigen determining sites also, so that it pulls up most of the antigens and forms a crosslink called lattice. Whenever a lattice is formed, this is rapidly phagocytosed and degraded by the macrophages. To speak little more about the antibody is Y-shaped molecule. As I said, it will have 2 light chains and heavy chains in them. The light chains and the heavy chains are connected with non-covalent and disulfide covalent bonds. The light chain is made up of 220 amino acids, and the heavy chain is made up of 440 amino acids. These the region where the upper region is called the variable region and the lower region is called the constant region or the FC region. Which there are many classes of antibodies based on the functions and the structural differentiation. So coming to the classes of antibodies, there are IgG, IgM, IgA, D, and E. So IgG, it is present in blood and tissues, and this is the only class which is capable to cross to the placenta. They secrete the molecules to the mother's milk, thereby developing immunity to the protection to the fetus and the fetus and the born child. So the IgM is the first class of immunoglobulin, which is produced by the B cells when they get mature. There are five. They form a pentamer molecule always with 10 antigenic binding sites and attaches to the outer coat of the bacteria. And then agglutination is the process which they use for killing of the microorganism, thereby giving protection against microbes. IgA is the main class found in body secretions. Like saliva, tears, colostrum will be having IgA. This is produced by the B cells, which is present in the mucosal membrane. So particularly they are defensive at the mucosal surfaces. This is not stable as IgG, and so hence it is very less, only low amounts are available in the mucosal surfaces. IgD, this is present in surface of most of the B cells. Whenever the B cells gets activated and get mature, it produces the pro-inflammatory cytokines. This helps in IgD helps in producing these pro-inflammatory cytokines, and the specific function is not that clear, but always helps or concerts with other cells to provide the immunity. So IgE it is present in some of the mast cells and basophils. So there is a release of histamines, and this IgE antibody helps in histamine release, which is the allergic response, and more symptoms like running nose, sneezing, etc. will be because of this IgE production at the inflammation site. So it is involved in local immune response communication. So this is a combination or a gist of whatever we have seen now. Innate immunity, which is the rapid response. Adaptive immunity is the slow response. So these are the cells involved in innate immunity, and these are the cells involved in adaptive immunity. But you can see there are two types of cells, which is a bridge between the innate and adaptive. So to know little more about this gamma delta T cell and the natural killer T cell. So this gamma delta T cell is very minor, a small amount of T cell subset. This bridges between the innate and adaptive. It mainly involves in cancer progression because they have the anti-tumor cytotoxicity property and strong cytokine producing ability. Though it is a very small subset, it involves in the cancer progression. So this gamma delta T cells has been recently used mostly in cancer immunotherapy. A lot of attention is there to research about these T cell and find out more exploration on this type of T cell. Natural killer T cell. You can see a natural killer cell will be involved in an innate immunity, but this is natural killer T cell, which has a combination property of natural killer cell and T cell. So this is called NK T cell. This expresses both TCR characters of adaptive immunity and surface receptors of B cells, NK cells in the innate immunity. So this involves in surveillance of tumors, maintains self-tolerance, and regulates autoimmune diseases. So what can be the key takeaway from this webinar is, like you would have been known that innate immunity is the first immunological response we'll be having whenever we are fighting against an infection. That will be a non-specific mechanism. And then adaptive immunity will develop in conjunction with the innate immunity. Only when innate was not able to fight Then comes the adaptive immunity to eliminate the infectious agents. But when on the second visit, adaptive immunity will have the memory, so it rapidly activates and gets into the action. So there will be development of immunological memory in adaptive immunity, and there is a synergy between this adaptive immune system and its innate counterpart. So in either of the system, if there is any defect, then it leads to immune pathological disorders like autoimmune deficiencies in immune system, hypersensitivity reaction, etc. So to just let you know the research hotspots in the immunology, there is more extensive research going on as the subject in itself is very vast and there are more to learn. There is a extensive research going on this in immunology from cells to molecular and to gene level, and the mechanism mainly involved in cellular and molecular of immune regulations and the immune recognition, which is structural based, and the related mechanism, inflammations, innate immune regulations, and the immunotherapy is again a very crucial part now. I mean, which has been a very supportive for more diseases nowadays, and the immune memory. So these are all the basic, basic research going on, and there are streams in this immunology like clinical immunology, diagnostic immunology, tumor immunology, vaccine immunology, and many more are there. This is just to give a gist on that. So this is the end of the webinar, and so this webinar has been conducted by this Sri Sai Clinical Research Institute, which has been designed mainly for training, workshop, and internship program, where you will get hands-on experience working alongside with industry experts also. And you also gain practical insights for whatever the training they are giving. So, and this has industry partners like Luminal Bioform, KMS, MTR, Workshare Clinical Research. Workshare Clinical Research is a full service CRO contract research organization which focuses on clinical research services across the globe, and it also involves in diagnostic services, project management, regulatory affairs. KMS Health Center Private Limited is the partner of choice to global pharmaceutical companies in the areas of novel drug delivery system development, custom pharmaceutical product development, R&D services, and also it supports end-to-end product development of complex and niche products. They also offer services like formulation development, analytical development, and tech transfers. And coming to Luminal Biopharma, it's an integrated biopharmaceutical company established to brighten up human healthcare, especially with biosimilar medicines, and socioeconomic focus with its own technology and product development. It offers full range R&D, analytical, preclinical, and clinical development services, and seek comprehensive and selective partnership and services. It also offers an unflinching support to the academics towards developing the future generation in biotechnology. So for any more industrial, this is just a glimpse of the industrial visit we had recently with the Sri Sai Clinical Research Institute. And for more industrial training, you can reach out us by contacting these numbers. For queries or clarification also, you can contact here. We are always happy to help you. So the session is open for QA. You can submit your questions, whatever in the chat box, so that we can try to respond in that. So the transplantation immunity, somebody has question. So the whenever a transplant occurs, like it can be an organ or any organ transplantation is happening, there will be an immune response at both cellular and humoral level. Like as we discussed, it might be of lymphocyte mediated and antibody mechanism. So there are so many steps to check whether it is accepting the graft or rejecting the graft. So based on that, whatever the immunity is developed is the transplantation immunity. Participants can fill up this feedback form with the link provided in the chat box so that you'll be receiving the certificates. So if any more queries are there, you can put in the chat box. When the feedback form is submitted, you can receive the updates on the other rest of the webinars upcoming, which will be communicated to you through WhatsApp. Hope there is no more questions, so that I think like we can wind up the session. Thank you so much, participants, and please fill up the forms as to get the certificates. Thank you.